Hi to Dr. Norashikin and my fellow colleague. My name is Muhammad Amir bin Muhammad Azman, student ID of 66788. Today, my group and I will be presenting on the effect of sucrose in in-vitro axillary shoot buds regeneration from nodal segment of Z maize. Today, I will be responsible in introducing to you guys what is Z maize. Z maize, which commonly known as maize, as you can see up there, is an individual from genuine grass family known as Poaceae. Z maize contain fiber, provitamin A, vitamin E, and rich in minerals, making it as an ideal for daily consumption and source of nutrients. Maize can be used as food in the form of flour, oil, sweetener, cereals, all the way it is. Maize can also be used as biogas. From the statistic, we can see that United States, China, and European Union are the top 3 countries with the highest number consumption of maize. And in Malaysia, we can see that consumption of maize has steadily increased annually. Over the past 10 years, consumption of maize has increased around 29.03%, which equivalent to 8,000 metric tons within the year 2009 to 2019. As you can see, Malaysians are consuming more and more corn year after year. Two of the major reasons or factor is due to the fact that 1. Corn is cheap. Second, it provides all the essential nutrients that we need. It is one of the go-to food for those who are from low-income family or those who are facing financial crisis. By studying the effect of sucrose in development of maize, we may one day be able to produce maize in shorter period of time to meet with the increasing demand of maize.
Hi everyone, I am Siaw Ying with metric number 66507. Now I am going to talk to you about the 2 problems that led to our research. The first main problem is slow conventional propagation of maize. As we know, the old conventional method practiced by farmers recently are unable to meet the increasing in the demand by consumers nowadays. The old method, which used seeds to plant the maize, tends to be affected by the environmental factors such as the undesirable light and temperature. This will lengthen the time by the seeds to germinate, and also when the seeds are germinate into seedling, they will tend to be affected by the several factors such as disease caused by the bacteria in the soil, and therefore this will affect their growth and development into mature plant. Therefore, we need tissue culture to propagate the plant in in-vitro environment, which is in a sterile condition. A sterile condition can provide a better template to grow the plant in a large scale, because in sterile environment, the plants would not be affected by the bacteria like the one present in the soil. In in-vitro culture of plant, the plants also supply with the nutritional requirements that it needs, and also with the light and temperature. Therefore, we need tissue culture so that mass propagation of the clonal mass seedling can be produced in a shorter time to meet the demand of consumers in lower stage. The second problem is there is a limited protocol that studies on the optimal sucrose concentration used by culture of nodal segment of mass. The available protocol, as we know, is only focused on the other parts of the mass, such as embryo or seeds of mass. The researchers study the effect of several sugars that affect the growth and development of mass embryo. However, there is no study available yet for the culture or study of the optimal sucrose concentration in culture of nodal segment. And also we cannot use the protocol in the culture of mass embryo on the culture of nodal segment because every part of the mass need different nutritional requirement for inducing their optimal growth.
Hello everyone, my name is Vanessa and my metric number are 67984. Today I'm going to present you the objective and the literature review of our proposal. So the main objective of our proposal are to observe the sucrose effect on the in vitro shoot regeneration of maize plant, which we call as the Az-zahra. So the sucrose concentration that we use are the lowest 0.25%, 0.5, 0.75, and the largest value that we use are 10% concentration. When the sugar concentration are increased, the shoot regeneration decline is high concentration of sucrose could inhibit the process. For example, the process that we say, the process are the photosynthetic key enzyme and the chlorophyll formation process. They could be inhibited if the sugar concentration are increased. The other main objective of our proposal is also we want to know the importance of non-reducing and reducing sugar used in the plant tissue culture. For example, the reducing sugar are glucose, fructose, ribose, and xylose. And for example, of non-reducing sugar are the sucrose. Sugar are used to induce the tissue culture differentiation. Sucrose are also used to supply energy source for sustained growth of the plant. So we know that sucrose are very important in the plant tissue culture.
In this experiment, we hypothesized that the number of axillary shoot buds induced will increase along with the increase in sucrose concentration supply in the media. However, when reaching the point of saturation, we hypothesized that the number of axillary shoot buds induced will show decline because too high osmolarity in the medium caused by the too high sucrose supply will restrict the roots from absorb nutrients from the media.
Okay, now we proceed to the next slide. That's our literature review. Plant regeneration. Plant regeneration are referring to the ability of plant that able to regenerate, if not all, but most of its organ, and can be enhanced exogenously by supplying with plant hormone, different type of sugar, and nutrition, and also the balance of auxin and cytokinin. There are two types of plant regeneration. That is in-vitro shoot regeneration and in-vivo shoot regeneration. So in-vitro are referring to the cultivation of isolation of maritime cell of the maize plant in a controlled environment. For example, in a culture media or outside a living organism. The thing that we can control it. And then in vitro are referring to the condition where an experiment is conducted within the organism of its natural condition. The thing that it happened naturally and we cannot control it. That's the difference. And then also the difference between in vitro and in vivo. So we can conclude that the difference between in vivo and in vitro are from the way the experiment takes place and its limitation due to its different properties. That's all for me. Thank you.
Assalamualaikum, I back to Dr. Norashkin. I am Maisarah Filza Az-zahra. So now we are moving to the methodology section. For methodology part, I have divided the methodology into 5 parts, which is explain, second, medium and cultural condition, third, hardening, fourth, acclimatization, and last is statistical analysis. I will explain the methodology section one by one. First part is explain. So in explain, we remove the high selected and disease-free in vitro seedling of maize from the culture media, and then we remove unwanted part. Then we wash the shoots of maize under running tap water for 25 minutes and then in the tipple for like 5 minutes. Then we rinse it with sterile DDW. Next, the surface sterile in mercury chloride or HgCl2 with 0.1%, and then we rinse again with sterile DDW. Dissect each of maize seedling into separated nodal segment and then transfer the excision shoot into half ms basal medium that containing 0.25%, 0.5%, 0.75%, 1%, 2.5%, 5% and 10% of different sucrose concentration and solidifies it with 0.8% agar. Okay, we are moving to the second part, which is medium and cultural condition. Okay, in this part, we use half ms basal medium of different concentration of sucrose for regeneration of shoot from the nodal segment of maize. Make sure the pH of medium to 5.8 to the addition of 0.8% agar and autoclave it at 15 lbs and temperature at 1 to 1 degree Celsius for 20 minutes. Keep the temperature at 25 until 27 degrees Celsius with 16 hours photo period under lux fluorescent light intensity. Next part is hardening. In hardening, we carefully take out the rooted plantlets from culture vessel and then we wash it under running tap water to remove remaining agar. After the agar removed properly, we dip it into 0.1% IAS solution for 1 minute and transfer it to tray containing sand. Okay, we are moving to acclimatization. For 1 week, keep the potted plantlets under transparent polythene membrane to ensure high humidity and then keep in open in diffuse light for hardening. After 7 days, move the surviving plants to pots containing garden soil and maintain in greenhouse for acclimatization. The very last part of methodology section is statistical analysis. In statistical analysis, we repeat the experiment 3 times and each treatment has 10 replicates. And then the purpose is for determine and compare the number of axillary shoot buds from the nodal segment on different concentration, and we observe the root form from the different concentration of sucrose. For this analysis, we use standard deviation to analyze the data for the shoot number per explant.
The proposed research is to provide substantial amount of evidence about the effect of sucrose on in vitro shoot formation of the maize plant. It is expected that the number of shoot formation will increase proportionally as concentration of sucrose also increases. Based on the study, the optimum concentration of sucrose is in between 0.25 to 1%. Using beyond 1% of sucrose concentration will cause declination in the shoot formation. This study will help the farmers and plant breeders not only to increase their crop yield production, but with less amount of time, lower costs, and much more efficient. All in all, it does not only boost the agricultural economy, but also provide resources globally. This study will help in further development of the study in shoot formation, not only in the maize plant, but also other plants as well. Thank you.